Okay, so first of all, I thank everybody for attending this presentation. This is work done at the University of Idaho in the Department of Biological Sciences, and it's essentially exploring the evolution of antifungal toxin resistance in the pathogenic species Candida glabrata. So before I really get into the science, I just want to thank essentially everybody who's been in my lab since I started my laboratory about 5 years ago. A lot of undergraduates have been involved directly in this work. It's actually primarily been driven by undergraduate research, and a lot of the funding has come specifically from the NSF, and of course a large portion of this is from Beacon itself. And I do credit Beacon with giving us the money that has actually supercharged this project and really made it the story that it is right today. The primary instigator of most of this work is Lance Fredericks. So he currently is a research technician in my laboratory. Thanks to the pandemic, he's actually going to be applying as an MD PhD student. He's actually applying as an MD PhD student right now. So I expect big things from him because he's really been a powerhouse in the laboratory. Okay. And if any time during this presentation anybody wants to ask questions, please just butt in. I don't mind stopping and going over things as needed. It's just quite different stuff from usual seminars. Okay, so a little bit of obviously background. So we're interested in fungal diseases, particularly pathogens that infect humans. Fungal disease is really a big problem, but it tends to be not as well thought of, despite the fact it's killing 300 million people. It's infecting 300 million people and killing about 2.5 million every year. And compare that to malaria, which kills about 500,000 people annually. We're thinking about 15% of healthcare associated infections are caused by fungi, so it's quite a significant amount. And Candida species of fungi account for the majority of invasive fungal diseases. And invasive fungal diseases tend to be those that are most serious because they're occurring generally in people who are immunocompromised or have existing health conditions. Some examples that you may have heard of are such as oral and vaginal thrush caused by Candida species of yeast, as I said, mainly Candida albicans, but there are other Candida yeasts, as you will see, that are important for causing these mucosal diseases. They can also cause invasive disease, which again is considered as more life-threatening. But there are other fungi too that cause these kinds of diseases, Candida being the major one. More topical, there is emerging fungal disease that's occurring all the time. So for example, people who have had coronavirus, particularly in India, there seems to be some association with mucormycosis, which is an extremely debilitating disease and causes a lot of tissue necrosis and deaths. And so that's been sort of an emerging disease that's arrived out of the coronavirus pandemic, but there have been repeat episodes of the emergence of fungal pathogens that have wiped out either specific species of trees. For example, the American chestnut tree used to be extremely prevalent on the East Coast and was completely wiped out by fungal disease. The problems we're having with amphibian populations currently and those being wiped out by chytrid fungus. So yeah, they're a big problem in general, but in humans, yes, we do certainly care about that too. So are we in need of new treatments? Well, I would say yes. We only have really 4 major classes of antifungal disease. For antifungal diseases, and most of them are actually toxic to humans. And that's not necessarily surprising because we're trying to think about drugs that will target fungal specific processes. And there are not that many, seeing as fungi are relatively closely related to humans, and we share a lot of the same biochemical processes. But in addition to the lack of antifungal classes that we actually have, there's been an increased prevalence of drug resistance in certain species. And some emerging opportunistic pathogens, such as Candida auris, are innately drug resistant right from the get-go. So drug resistance is a big problem, especially because we have limited therapeutics. And of course, the increase in medical procedures and the ability to treat people that are very sick due to altering their immune systems and damping down autoimmune disease and such, we can have immunocompromised populations that are particularly at risk from fungal disease. And that also includes pregnancy being an immunocompromised state that can cause a prevalence in certain types of fungal diseases. So yeasts, whether or not this audience is familiar or not with yeasts, I'll give a brief introduction. Single cell microorganisms, they're a type of fungus. Okay, example of an EM of Saccharomyces cerevisiae, which is the Latin name for yeast that we essentially use to make all of my favorite products, and probably some of yours too. Okay, we've got obviously fermentation to make wine and beers, to make bread, but also fermentation by Saccharomyces yeast is linked, or is extremely important for the production of cacao and coffee as well. So really, really important from that point of view. But of course, speaking to this audience, we also know the importance of Saccharomyces cerevisiae as a model organism to study fundamental biological processes. But we're not going to talk about fundamental biological processes from that perspective in yeasts. We're talking about killer yeasts. Now, killer yeasts, it's quite a scary thing to say, talk about killer yeasts. Are we thinking about pathogens here? We're not. We're actually thinking about yeasts that naturally produce antifungal proteins that prevent the growth of other fungi. Now, I didn't coin the term killer yeasts. It was actually first coined way back in the 1960s. So it's not my fault that they're called killer yeasts. But they produce these antifungal proteins that are generally referred to as killer toxins. And here's an image of what a killer toxin would do if it was a yeast that was producing a killer toxin would do if plated in competition with other yeasts. So on the left-hand side here, we have a killer yeast that's been spotted in the center, and it's producing a toxin which is causing the inhibition of a competing yeast that's been spread at a lower density on an agar lawn. So you can see these robust zones of total growth inhibition around the killer yeast. Similarly, if you do the same thing with a non-killer yeast, obviously we don't see those large zones of growth inhibition because they're not producing this killer toxin. So there are currently 9 different types of killer toxins produced by Saccharomyces species, and that's including laboratory strains of yeast that are commonly used to study basic biomedical processes, biochemical processes, such as SK1 for those of you that work with yeast. So what can we do with killer yeast? Well, we can make beautiful pictures. And I like to tell people that I did this before I became a faculty member, as I was a postdoc, because I have no time for this kind of thing anymore. But you can essentially see the same kind of thing. You've got the killer yeast has been drawn as these icosahedral shapes. And you can see that they're producing toxins, which are inhibiting the growth of this lawn strain to create these beautiful patterns. Okay, but more seriously, can we use killer toxins to treat diseases caused by fungi? This was really the central motivation for studying killer toxins in the 60s and the 70s, mainly because at that time, when killer toxins were first discovered, there was only a single treatment for invasive fungal disease, and that was amphotericin B, which is still used today. Highly toxic antifungal drug, but still quite effective. So there was this idea that you could use killer toxins as a new form of antifungal therapy. But of course, we have some issues with using killer toxins, and it became very apparent early on that when you have a killer yeast that produces a killer toxins don't tend to be broadly antifungal. Okay, they're not like bleach or any kind of chemical compound that would broadly inhibit all fungi. Okay, they are extremely strain and species specific, and that's demonstrated here. And you'll see more of these kinds of graphs as we go through the presentation. So we have killer yeast 1, and you can see that these yellow boxes indicate strains of yeast written down here that it's not inhibiting, but it quite effectively inhibits a lot of these ones on the right-hand side, indicated by these darker blue colors. And the dark blue colors, we're just sort of using this qualitative assessment for a degree of growth inhibition, where dark blue means we get these large zones of methylene blue staining, which is indicative of cell death, but also zones of growth inhibition too. But you can see that this other killer yeast 2, has a different spectrum of antifungal activity. And if you really look closely, you can see that we've got strains of cerevisiae, Saccharomyces cerevisiae here, that are susceptible to these 2 killer yeasts. But then down here, we've got another strain of cerevisiae that is completely resistant to these 2 strains of killer yeast. So of course, if you're thinking of an antifungal drug, you don't really want to have strain specificity when you're just trying to remove a pathogen from an individual. You don't have to want to do strain typing to choose which killer toxin to use. It's not very appealing from that point of view. But students can be quite persuasive, can be very persuasive. And essentially, when I started the laboratory, I had no intention really of working on pathogenic organisms. But they really did. And even though I thought I knew what the answers would be, we ended up persevering and looking at how different killer toxins can affect the growth of opportunistic pathogens and professional pathogenic yeasts. And so this work, again, as to reiterate, was mostly done by undergraduate researchers. And we use biosafety level 2 precautions in my laboratory, just because some of these pathogens are, like I said before, extremely drug resistant. And you never know when you're immunocompromised. So we do treat these organisms with respect. And that's actually quite a high bar to set for an undergraduate researcher. So I credit all of these individuals with working really hard to perfect the techniques and working safely in my laboratory to study these pathogenic yeasts. And that started way early on with Emily Kaiser. And like I said before, has been really pioneered and pushed forward by Lance Fredericks, as well as help from other undergraduates. So this work was actually published this year. And so we did the screens that I suggested. We looked at how susceptible killer yeasts are, how susceptible pathogenic yeasts are and opportunistic pathogens are to different killer toxins produced by Saccharomyces yeast. And so I mentioned there was a number of different killer toxins, and they're written down here on this graph at the bottom. And we see that even the best killer toxins only kill between 10 and probably 18% of pathogenic yeast that we assayed. Okay, and we actually used Candida yeasts. They were our main target because they are the most important for nosocomial infections, so hospital-acquired infections. So we tested 16 different species that are available from the CDC, FDA, and the USDA. And so this sort of vindicated what I thought all along and what is written in the literature, that killer toxins aren't broadly antifungal against different species of yeasts. But that's not saying that they didn't inhibit anything. They certainly did inhibit some of these pathogens. If we break it down by individual Candida species, we again saw situations where the vast majority were not susceptible to any of these killer toxins, and they're not shown on this graph because we tested 16. Some of them are susceptible to only one type of killer toxin. But we did notice that Candida glabrata did seem to be susceptible to all but one of our killer toxins. So you can see what Candida glabrata susceptibility looks like here. Obviously, it's not as susceptible to all the different killer toxins. It's not the same susceptibility. So some produce large zones of growth inhibition, and some produce only very, very small zones of growth inhibition that are barely perceived on the plate. But you can clearly see that we've got this one killer toxin here, this K62, which is actually not inhibiting Candida glabrata at all. And that's a completely different project that I don't have time to talk about today, but it's something we're actively investigating. But so at least it did seem like Candida glabrata seemed to be susceptible to killer toxins. So that was nice. And then we forgot about it for a little while. And then we decided to go and look at other killer toxins, right? So and seeing whether we could find new killer toxins out there by just surveying yeasts for toxin production. And so this has been obviously one of the easiest things for undergraduates to do in my laboratory. And that's essentially spread lawns of yeast, spot on potential killer yeasts, and look for zones of growth inhibition. And we've done this extremely effectively, initially starting using the typical 9-centimeter plate and spotting the yeasts and looking for the production of killer toxins, which is quite obvious in this case. And we screened a lot with this very, very simplistic method. And then we graduated up to using 96-well pinners, manually pinning arrays of different yeasts, and again, looking for the killer phenotype, zones of growth inhibition. And then we graduated up even further to start using robotics, which was funded by, which was kindly provided by the Institute for Modeling, Collaboration, and Innovation here at U of I. So I credit them for purchasing this instrument. And again, it's been a game changer in terms of the throughput of the assays that we can do, because this machine will print from liquid to solid, solid to liquid, liquid to liquid, solid to solid. It'll do anything we want. And for that, I think even this number is out of date right now, 1,600 yeast. I think we've tested more, and more than 23,000 interactions to look for novel killer yeasts. And so ultimately that's led to, we found a lot of killer yeasts. 22% of all that we've surveyed in Saccharomyces cerevisiae produce killer toxins. And that's particularly important for a lot of people that are studying, how different yeasts interact with each other in different environments. I know of evolutionary studies where people mix yeast together under different conditions and see who wins. But they typically don't consider that a lot of these yeasts are actually producing these offensive chemical weapons that would certainly skew any of these kinds of in vitro competition assays. So that's sort of an aside, but still an important thing to mention. So we have now this larger collection of killer yeasts. And so we thought, well, okay, we knew that glabrata was somewhat more sensitive to killer toxins than any other yeast. So we did a bigger screen, looking at more strains of Candida glabrata, because remember I said that individually, strains of yeast within even the same species can have different susceptibilities. And so we thought, okay, well, we knew that the type strain was susceptible to most killer toxins. So we then took a collection of 133 different Candida glabrata strains from literally all over the world. We didn't travel, but we just ordered them and begged, borrowed, and stole from other laboratories. And we came up with this really nice, what we feel is a representative collection of the global diversity of Candida glabrata. And then we hit them all against a collection of killer yeast, which we thought were pretty good killer yeast. So the killer yeast are on the side here. And so we're looking at the strains of glabrata coming down in the columns. So what we see is that we've got this group of killer yeasts, which to all intents and purposes can inhibit every strain of Candida glabrata that we challenged, that we were using as to challenge them. So that actually was extremely unexpected, because normally when we used Saccharomyces yeasts and tested large populations of Saccharomyces cerevisiae, we would get patterns that would resemble sort of the bottom half of this cluster diagram. Killer yeast would kill a few, but generally speaking, there would be large populations of yeast that were resistant to these killer toxins. So we were extremely surprised to see such good growth inhibition by a smaller subset of killer yeasts. And of course, that led us to think, well, what type of killer toxins are they? And so that's why we're going to shift our focus just a little bit, and tell you that the majority, not all, but a lot of killer yeast, Saccharomyces killer yeasts, are persistently infected with double-stranded RNA totiviruses that enable toxin production. So you need infection with these viruses to enable toxin production. That's really important. But it's not the totivirus itself that actually encodes the toxin. Okay, so this is where it gets a little bit more complicated. There's actually a satellite double-stranded RNA that parasitizes the virus to enable its own replication. And the killer toxin genes tend to be associated with these double-stranded RNA satellites. So this actually, even though it's quite complicated that you've got a virus and you've got a satellite that encodes a toxin, it actually makes life so much easier for the discovery of novel killer toxins, because we can selectively purify the genomes of both totiviruses and, if they are present, any accompanying double-stranded RNA satellite. So you've seen this graph with this representation before, a killer yeast and a non-killer yeast. What we'd expect to see in a system that's dependent on a double-stranded RNA virus and satellite is that when we take cell lysates of yeast, when we purify the double-stranded RNAs using cellulose chromatography, we see distinct bands that are representative of the double-stranded RNA genome of the totivirus and the double-stranded RNA genome of the satellite. And they are quite easily resolved on a standard agarose gel. On a non-killer yeast, we would either expect there to be no double-stranded RNAs or only the presence of a totivirus. Okay, so we need the presence of this double-stranded RNA satellite to essentially produce the killer toxin. Okay, and of course there are nuances to this, and I'd be happy to talk about that in more detail. But we'll just keep with this this more simplistic interpretation. Now, of course, again, killer toxin plate assays, brilliant for undergraduates. Cellulose chromatography sounds scary because I said the word chromatography, but actually it's not at all. Undergraduate researchers have again led the charge in actually doing cellulose chromatography using very, very simple lab equipment to actually be able to extract double-stranded RNAs from yeasts very effectively. So this has been, again, another thing that's been driven by undergraduates. But of course, we've got double-stranded RNAs. We want to know what toxin genes are present on them. So this actually was a separate side project that, again, we published a few years ago, but it was actually developing a method specifically tailored for Saccharomyces-derived double-stranded RNAs and how we would actually sequence them using Illumina sequencing. And briefly, what we do is we take the purified double-stranded RNA, we actually give it a poly-A tail, so then at that point it looks more like a messenger RNA. And then we run through standard protocols of messenger RNA, like transcriptomic type amplification through using an oligo-DT oligo. And this one has an anchor sequence on it to actually help it be more specific. And so that cDNA creation can then be used to essentially create full double-stranded cDNA copies of the viral double-stranded RNA. And so this work was again supported by the generous contributions of Idaho Imbri and also what was IBEST and is now the Genomics and Bioinformatics Resource Core. And I always have to apologize them because we were actually able to break an Illumina sequencer. If you're ever going to polyadenylate anything, don't add too much poly-A polymerase because then all the sequencer does is sequence A's and the run will crash. So I always apologize to Barry Roberson for breaking the sequencer at that time. It was totally fine. And again, this work was spearheaded. In this case, it was a little bit more advanced. This was spearheaded by my research technician at the time, Angela Crabtree. So now we were able to sequence our double-stranded RNAs, and so we would get these contig graphs that would essentially very clearly show the presence of totiviruses that have long lengths and high coverage, and then also the satellite double-stranded RNAs as well. So very, very clear to resolve the viruses and the satellites that were present and responsible for toxin production. Okay, so this is a lot more straightforward than prospecting through whole genome sequences looking for novel killer toxins. It makes our life somewhat easier. So we come back to our initial discovery. Candida glabrata seemed to be more susceptible to a group of killer yeasts. And so all of these killer yeasts had virus satellite systems, and we were able to successfully sequence all of them. And so they turned out to all be a killer toxin called K1. Which is actually the first killer toxin ever discovered. So we were a little bit deflated that we didn't actually find any novel killer toxins for this activity. But we did take this unbiased approach, and we did notice that it was the K1 killer toxin that was responsible for the inhibition of Candida glabrata. And that then, at this point, pushed on the rest of the projects. So a little bit about K1, just to give some background. K1 is obviously it's an extracellular protein toxin that initially binds the Saccharomyces cerevisiae or other yeast cell wall, typically binds the 1,6-glucans in the cell wall. And after binding the cell wall, will interact primarily with the Cre1 membrane receptor. And it's thought that the Cre1 membrane receptor somehow enables the formation and the attack of the cellular membrane by the K1 toxin. And it's an ionophoric toxin because this attack by K1 leads to the efflux of potassium and we think also hydrogen ions from the yeast cell. Now, this ultimately leads to cell death, either through programmed apoptosis at low concentrations of toxin or full-on lysis at high concentrations of toxin. So just remember that you've got this important role for the Cre1 GPI-anchored membrane receptor. It's an important player that we'll talk about a little bit later. Okay, so we obviously wanted to prove that K1, it wasn't just a feature of killer yeasts. So we've worked on protocols of being able to purify K1. So we've got this very straightforward precipitation protocol, which we can use to partially purify K1. And we've raised antibodies to K1 as well. So we can actually detect it in culture supernatants quite effectively. So we have the tools to be able to now assay somewhat purified killer toxins and look for their effect. So we can see that represented here. We can see this nice concentration-dependent growth inhibition of this lawn yeast by purified K1. And it essentially recapitulates exactly what we saw with the killer assays. So we see that if we take 30 nanograms of purified K1, it will inhibit every single strain of Candida glabrata that we tested it against. So it does suggest, even though we had this far side of the graph, some of these killer yeasts weren't effective at killing every strain, but when we did expose them to 30 nanograms of purified K1, we did see robust inhibition of all strains. But of course, some strains are more susceptible than others. There's some that are extremely susceptible and some that are certainly less susceptible, but every single one is susceptible. Which again, remember, like I said, that's a really big deal. That's not something that's really been thought of before. Okay, so last thing I'll mention about this is that acquired drug resistance is a big problem with Candida glabrata. And one thing that we were worried about was that, well, okay, what if we've got what if K1 resistance, antifungal drug resistance is correlated with K1 resistance? Well, we already knew that the strains that we tested, the glabrata strains we tested, were not resistant to K1, but some were more resistant than others. And we were wondering, does that track with inhibition by other antifungals that are important for use in clinical studies? So this is an example of meconazole, looking at the minimal inhibitory concentration of meconazole. Yeah, sorry, it's both MIC. It's a little confusing. But essentially what we see is that, the different MIC values of these different Candida glabrata strains, they don't correlate with the degree of growth inhibition that we see with K1. And overall, that plays out for all of the ergosterol biosynthetic inhibitors, all the azole antifungals. We don't see any correlation there. If we've got the base analog, 5-fluorocytosine, we don't see any correlation there. With the ergosterol binder amphotericin B, we do see that there does seem to be a slight correlation. But again, there is none of these strains were completely resistant to K1, but we do see that slight correlation, which again is important later on. And again, this work was in collaboration with Dr. Jack Sobel, a real towering expert in the field of Candida glabrata, vulvovaginal candidiasis. So we're grateful for his collaboration on this work. So why do we care about Candida glabrata? I just want to emphasize it a little bit. Candida and vulvovaginal candidiasis is a really important issue for women's health. 3 out of 4 women will suffer vulvovaginal candidiasis at least once in their lifetime. And typically, we think there's about 12 million women in the USA that suffer from at least 4 infections of vulvovaginal candidiasis every single year. And that's classified as recurrent vulvovaginal candidiasis. And so Candida glabrata can account to up to 50% of these recurrent infections. And honestly, I just told you before, Candida glabrata and many other Candida yeasts are either gaining resistance or have intrinsic drug resistance. So we don't have effective therapeutics to actually help this significant number of women that are actually in pain for a lot of their lives because of these recurrent infections. So is then all the work that I've shown you previous to this is showing you that K1 can kill Candida glabrata. So is it a possible treatment for VVC? Well, we've got this optimal activity at pH 4 to 4.5. It's broadly inhibited to Candida glabrata. It's active in conditions that we found simulate the vaginal mucosa. That's shown here in this media experiment here under hypoxic conditions. But the thing that we show that it's non-toxic to human cultured cells, which is shown here on the bottom. So everything looks good here, but the one con we have is that it's inactive at 37 degrees. But that's something that we've recently addressed, and we find that we have found a compound that will stabilize K1, and that's been really important for a lot of the assays we've been recently performing in our laboratory. The ability to stabilize K1 at 37 degrees is a real big game changer. So we certainly hope to utilize this in the future. But of course, this is a beacon seminar. You're all wondering where the evolution is. Okay, well, I've given you all of the background, all right? And of course, been showing you all the cool work we've been doing in the laboratory. But now I'm going to get to the point where we really think a little bit more about, well, why is Candida glabrata susceptible to K1? Okay, if we look at related species of yeast, we find that glabrata is uniquely susceptible to K1. There is no other species that we've tested that has this broad susceptibility to K1. Now, one caveat is we don't have the large strain collections that we have for any of these other species, these exotic yeast species. But the main point is that we've got these somewhat closely related yeast species, Candida yeasts, that actually are also opportunistic human pathogens, just like Candida glabrata. They're not as prevalent, but we see that they are resistant to a lot of K1 killer toxins.
Do you mind if we ask some questions?
Yes, of course.
I just wanted to try and make sure I'm understanding the scope of some of this. Mm-hmm. And in particular, so at the beginning, I think you gave a number, and I could have be misremembering, but it was something like 250 million people a year are infected by some form of. Was it just fungal?
That's just fungal infections in general. So that can be anything from aspergillosis to candidiasis. So any kind of fungal infection. And they can be from non-life-threatening to life-threatening. Ringworm, for example, is a fungal infection. Okay,
but then you were also saying that 3 out of 4 women at some point in their life are infected with C. glabrata.
Yeah.
And I mean that obviously is a much larger number, though, over entire lifetime, not just per year. And I mean, and I guess. Which it does seem like this would then have to make up the majority of the 250 million infections a year for all those numbers to work out, although maybe not.
Right. Well, so it's an interesting point because a lot of the cases, so a lot of infections are recurrent. The reporting of this, we obviously we actually think it's probably underreported a lot of the times. People don't like, and a lot of these are extrapolations to extrapolate over higher. So there is a lot of error in these numbers. But if I was to sort of like hang my hat on any particular number, I would say these numbers are considerably larger than what I'm giving here, mainly because a lot of these go undiagnosed. People suffer with them. It's obviously not a pleasant thing to talk about, even to a physician. And typically speaking, these kinds of women's health issues have been routinely underrepresented and ignored by the medical community. If males had similar symptoms to women who have vulvovaginal candidiasis, I guarantee we would have a treatment right now. And that, I mean, I know it sounds funny to say that, but really that's the opinion of a lot of people that work in this.
That obviously is true about many aspects of medicine.
Right.
But it does seem, though, like this would presumably be one of the largest issues to be tackling then. But I mean, in particular, the glabrata. Yeah,
glabrata, because it's drug resistant. Candida albicans can in most cases makes up the majority. Okay, but albicans can be effectively treated with a lot of azole fungicides. There is some resistance that can be that can occur with Candida albicans and can evolve, but generally speaking, the resistant strains are the non-albican strains. Particularly, like I say, is glabrata. But again, it's very population dependent. There are certain populations in Africa and India that it's like I say, approaching 50% and can be, in some single case studies, can be over 50% that are glabrata. But in other cases, it can be down in the teens.
Right.
So it depends on the population, depends on the cohorts, etc. So
okay, that all helps a lot with the perspective. Thank you.
No problem. Great questions. I appreciate it. Keeping me honest with my numbers. So it's definitely intriguing as to why Candida glabrata is uniquely susceptible to killer toxins. And so the question, of course, comes, well, can glabrata ever become susceptible to K1 as a killer toxin? Now, those of microbiologists and evolutionary biologists know, of course, life tends to find a way to circumnavigate any kind of problem. And so that's something we wanted to test. Does glabrata have the ability to evolve resistance to K1? And so, of course, now we've got the tools, right? We have the purified killer toxins. We have identified the killer toxin. We know that it's K1. So we were able to take Candida glabrata and expose it to these enriched cultures, enriched preparations of killer toxins. And what we tend to find, we would plate typically about 1 million cells on a plate after growing them for a few days prior to plating to generate some diversity within the population. And then we would wipe out the vast majority on the plate with the K1, but we would then see killer toxin resistant strains, or at least, not going to say killer toxin resistant, survivors come back up on the plate and grow, and we were able to pick those those particular resistant mutants of Candida glabrata, and then look at them further. And that was with the indispensable help of Maitreya Dunham over at UW. Her group was responsible for doing all the genome sequencing on the strains of Candida glabrata that we isolated. Even though she'd never done Candida glabrata before, her lab, just obviously worked really well and got us all the data that we needed for this, and have been a valuable collaborator on this project. So we were fortunate that most of the strains that we focused on only had mutations in a relatively few number of genes. We had a double mutant here, and we had 3 single mutants in genes that either had been previously known to cause K1 or K2 resistance previously in Saccharomyces cerevisiae. But we also found one gene, tau3, that had never been reported as being K1, causing K1 resistance, mainly because in Saccharomyces yeasts, the laboratory strains, it's an essential gene. So we did find, of course, these were all new for Candida glabrata, and typically these were all discovered in cerevisiae through the deletion collection, and we clearly don't have the deletion mutants here. We have point mutations, frame shifts, and premature stop codons occurring. So definitely a little slight different flavor. And of course, we found the tau3. So just looking at the resistance phenotypes, it's not completely resistant, okay? It's not total resistant to killer toxins. If we plate the resistant strain as a lawn and we put down a K1 killer, we see that the zones of growth inhibition are significantly smaller. Okay, so that was sort of the resistance phenotype that we see. So it's not total resistance. Now, if we look at purified toxin, we see the same story. We see that they do seem to be more resistant, but not totally resistant. And when we compare that to wild type, yeah, it does seem to be significant. Now, if we look at spheroplasts, so whole cells, of course, have an intact cell wall. If we look at spheroplasts, so in other words, cells that lack their cell wall, we do see that the strains still maintain killer toxin resistance. Now, that's particularly important because we're considering where the resistance phenotype is acting, okay? The spheroplast resistance says that it's something intrinsic to the cell membrane that is causing K1 resistance. We know that K1 attacks the membrane, and the fact that spheroplasts are also resistant to K1 say that there's been some fundamental change in the membrane that is resulting in this resistance phenotype. So we looked a little bit more in detail, and so we did the usual thing. We did growth assays. And in all honesty, we were pretty underwhelmed with the response. We thought we'd have major growth defects, but that's not something we saw. We did see a little bit of reduced growth on YPD. So that's a rich yeast media on agar plates. You see a little bit, but no obvious increased mortality when growing them, right? Mortality was, the viability was about the same, but there was a slightly slower growth rate in agar. And in liquid culture at either 37 or 25 degrees, we didn't really see any significant difference in the doubling times. So, typical growth rate assays did not flag these as being as there being any trade-offs due to killer toxin resistance. But when we started to look at other stressors, such as Congo red and calcifluor white, which are important cell wall stressors, we did start to see some phenotypes, although not very clear. Particularly for the Congo red was a really important was one that we did see a significant phenotype. And then if we look at increased sodium chloride in the media, we did see some more phenotypes there. But the most striking phenotype was resistance to STS, okay, a detergent. So again, suggesting that we had some changes in the membrane, right? So the Congo red phenotype is showing changes in the cell wall, okay? But we do see, again, this really big change in STS susceptibility, suggesting that the cell membrane is again being perturbed in a way by these mutants that are resistant to K1. So what else did we look at? We did look at adhesion, okay? And we did see that on polystyrene, adhesion is also significantly reduced. But we didn't see the same phenotypes necessarily with epithelial cells that we did in tissue culture. So we do see some differences in adhesion rates with these K1 resistant mutants, but importantly, we did see a phenotype on polystyrene. The last phenotype that we tested was looking at virulence. Now, even though Candida glabrata is an opportunistic pathogen of humans, it is a virulent pathogen in the model of fungal infection, Galleria mellonella, or larvae, which is a wax moth larvae model. And we published a paper in 2020 showing a new method for actually injecting wax moth larvae that is actually safe. Again, because undergraduates could then do it. Previous methods had you literally holding the worm between finger and thumb while injecting pathogens, which we felt was not acceptable for undergraduate research. So we actually developed this, well, I didn't do it, my students developed this nifty little device, which is essentially a pipette tip cut in half, and then we jam the worms in it and they hunker down in there and allow effective injection. And this is a GIF on loop. We don't inject them multiple times. It's just a one injection. So after injection of type strain of Candida glabrata, typically we see signs of an immune response, which includes melanization, lethargy in the larvae, and ultimately death after a few days. And so, of course, we tested this using our K1 resistant mutants. And as the title says, we saw a significant drop in virulence. Now, we didn't see total survival. They all did die eventually, but there was the death was significantly significantly later than wild type, which typically kills them all off after about 5 days. With the mutants that we tested, we typically see them dying all off, or not all of them are dead by 7 days. Okay, at which point we terminate the experiment. So all in all, we show that we don't have really strong growth phenotypes, but we do see evidence of growth phenotypes, of phenotypes in virulence, adhesion, and things to do with cell wall and membrane stresses. So that leads us to our model of what K1 resistance actually looks like in Candida glabrata. So we have this situation where we have something called the Cre1 receptor present on the cell surface, which I talked about before. And Cre1 is not alone in the cell wall and cell membrane. It actually tends to, we think it clusters in sterol-rich lipid raft domains in the cell surface. And that clustering, we think, is important for interaction with K1. So we think that's important for K1 activity. And so what we did actually want to do is actually confirm first that Cre1 was important in Candida glabrata. And so we took wild type Candida glabrata, and we find it's, of course, very susceptible to K1. And now we take a mutant Candida glabrata that has a Cre1 deletion, and we find that it is completely resistant to K1. So it seems to have a similar story to Saccharomyces cerevisiae. K1 is essential for Cre1 susceptibility. Sorry, K1 Cre1 is essential for K1 susceptibility in glabrata. And so that led us to test, well, what about lipid raft integrity? If we can disrupt the lipid rafts that we think is where Cre1 resides, if we disrupt those lipid rafts, do we also see killer toxin resistance? So we can use the chemotherapeutic adelphazine in yeasts. And when we don't treat with adelphazine, we see, again, this concentration-dependent inhibition of Candida glabrata. So we get these large zones of growth inhibition. If we treat with adelphazine, we essentially see a marked reduction in susceptibility to K1, in the fact that when we treat with 14 and 6 nanograms of purified K1, the cells don't care if they've been treated with adelphazine. So saying that disruption of rafts also causes K1 resistance, which is, again, sort of is supporting a model that lipid raft disruption and Cre1 are important in Candida glabrata. So, so we've got this idea now that Tau3, VPS53, and the double mutant are somehow interfering with the formation of lipid rafts in Candida glabrata. And it's this loss of raft integrity that is causing K1 resistance, is in a similar way that we showed with adelphazine. So that idea that, again, we would be losing both the Cre1 receptor for K1, but also it fits with also the view that we saw that there's less adhesion of these mutants as well on polystyrene. And a lot of adhesins produced by Candida glabrata are also resident in lipid rafts. So it does sort of fit that, that model. So a little bit, just a tiny bit, just to polish this off, Tau3, how is it linked to lipid raft formation? Well, it's not really been explicitly demonstrated, so it's new information. But what we do know about Tau3 is like a scaffold protein, and it's essentially regulates phosphorylation of CBK1 and KIK1 in yeasts that essentially lead to a phosphorylation cascade for ACE2, which is a transcription factor phosphorylation, and SEC2 phosphorylation, which is important for the regulation of the exocyst. And the exocyst is essentially an important complex that's involved in getting the fusion of membrane-bound vesicles to the plasma membrane. And this has been proposed as being very important for active cell wall and cell membrane construction during yeast budding. So we think that these Tau3 mutants are disrupting SEC2 mediated exocyst regulation, and therefore interfering with membrane and cell wall construction, which would knock on to membrane raft formation. Similarly, the GARP complex is where the VPS54 mutant is, and that is known to have impacts on membrane sterols and sphingolipids, which are actually really important components of lipid rafts. So both of these mutants do seem to fit with that idea of lipid raft disruption. And so we're not quite there yet with this story. I really hope we can get this finished within the next 6 months, and my students are working super hard to get this done. So we've got a little bit of preliminary data that we literally got yesterday. And we're doing a little bit of microscopy and looking at the distribution of membrane sterols in yeasts. And in Saccharomyces, in Candida glabrata. And on the left here, we have wild type staining by this ergosterol sterol specific binder. And we can see that obviously the ergosterol is membrane present. And then we look at the Tau3 mutant, and yes, we do see membrane staining, but what we notice, and it is somewhat subtle, right? These mutants aren't that sick in terms of growth rate. They're reasonably happy with these mutations. But what we do see is that we see that there seems to be more localization of the ergosterol to specific areas of the membrane, right? There seems to be these more compact foci in the membrane. And there does seem to be more internal staining of ergosterol in these mutant yeasts. Now, we can't draw any firm conclusions about this yet, but it does show that potentially there is disruption of sterols in the yeast membranes, which does fit with our model of lipid raft disruption and Cre1 disruption. And then the most challenging experiment that we're currently doing, which will sort of like cap this off, is we're trying to enrich lipid rafts using density centrifugation and seeing if we do see any changes in Cre1 localization to the raft fraction. So that they're literally the last 2 experiments that we've got to do before we actually hopefully publish this in a super high impact journal, Beacon cited as a supporting entry. So with that, I'd like to thank everybody. I'll leave the picture of the model up. If anybody has any questions about the work, I'd be happy to answer them. But again, thank you for your attention. I appreciate it. Thank you for the applause.
Thank you for the really cool story you just told us.
No problem. Thank you so much.
People should feel free to unmute themselves and ask questions, or they can type stuff in the chat. Either way is fine. We're a small group today.
Hey, Paul, could you say some more about the 37-degree stabilizer business? That's pretty interesting. Is it something, it's a generalizable principle or kind of something unique for this
So
particular
the problem
model?
is I can't really talk about it in too much detail.
Oh, no, okay, sorry.
Transfer will get angry at me. Because we've got a provisional patent on K1 as a treatment for VVC. But I mean, what I can tell you about it is that, you know, what we did when we first started working on this was that we knew that K1 was really unstable, and it's honestly been something that's plagued the literature. People always say K1 sucks, essentially. It really doesn't behave very well. You can't purify it. But what was told in the literature was that if you add yeast extract to the buffers, it's totally fine. And actually, that's even more complicated than it seems, because if we have yeast, if we have yeast extract in the growth media, we get toxin production. If we take away yeast extract in the growth media and just grow in a simple media, we actually don't get toxin production. We don't see it in the media. Something we're working on. I don't know anything about that. But so the separate issue is that when we take, let's assume we're growing it in rich media, which of course contains yeast extract. If we precipitate the toxins in that media and suspend it in a simple buffer, we get this above phenotype, right? We get no stabilization. What we found is, though, that if we put back the rich media, we get stabilization. Not very good stabilization, but we got stabilization. And then that led us to essentially unpick everything that's in YPD. Well, three things: yeast extract, dextrose, and peptone. Peptone, hopeless, doesn't do anything. Dextrose, a little bit of stabilization based on compared to buffer. Yeast extract, as you might have expected, yes, give robust stabilization, but not as good as compound A, which I cannot talk about. But those of you with a brain between your ears will probably figure where our train of thought would lead after following the literature trail and yeast extract. Okay, so yeah, that's all I can really say.
No, that's okay. I'm glad to hear my intuition at least was right, that it is pretty interesting. So. Oh
yeah, it is. It's great. But I mean, for me, this blew me away. I mean, my students are all so modest about their achievements, but people have struggled with this for forever, right? People couldn't purify K1 because it just doesn't. They couldn't stabilize it. But we have something now that literally we can do animal tests now. We have a collaborator down in Tennessee. We're hopefully going to be doing some mouse trials with Candida glabrata to see whether we can, administer glabrata, set up an infection in a diabetic mouse model for VVC, for vulvovaginal candidiasis. And we're actually going to use K1 as a therapeutic to cure these mice, which I'm so excited about, to see whether we can actually get this animal model working. So yeah, with this compound A, I'm really hopeful we've stabilized it to a point where we don't have to worry about body temperature. We just have to worry about all the proteases and everything else that's in the vaginal mucosa. But that's a different problem. At least it's one less thing for us to worry about.
So I'll start by saying I don't know how reasonable of a question this is, but in terms of the whole applied evolution side of Beacon and all of that, how hard would it be to look at how well the K1 is working and actually select organisms that produce a more effective K1?
Great question. I would love to think of experiments these are hard experiments. I've thought about it for a long, long time, and I believe actually it was one of my proposed objectives in the Beacon grant, in the Beacon grant that I proposed, and it's just really hard. From an evolutionary perspective, having satellite double-stranded RNAs makes sense if you want to rapidly evolve novel toxins, because they're multi-copy and they're replicated by an RNA-dependent RNA polymerase, which is error-prone relative to a chromosome. So it makes sense that you could push the evolution of K1 to be better. It's just that it's the assay system that we'd use to evolve that kind of toxin, and how you would select and pull out those mutants from that study. It's a question that I haven't been able to grasp very well. Some sort of serial passage mode, and serial challenge with something that's maybe a little bit resistant, but we've tried it for 37 degrees, but we end up just evolving yeast that are just better at producing toxin at 37 degrees. The toxin didn't change. So it wasn't the toxin that changed, it was the producer strain that changed. So you've got elements of copy number that can arise, and just general production of the protein that can arise too. So yes, I'm totally on board with you. This is one of the most interesting things I could think about to evolve new killer yeasts, but it's really just a hard question of finding a good experimental system to test it. Somebody suggested emulsion droplets and looking at things like that. And there's a lot of things you could potentially do, but not something we've tried.
So producing more at 37 degrees, is that at least helpful?
It was modest at the end of the day, and we think it was more to do with, again, it was more to do with the fact that Saccharomyces cerevisiae just grew better at 37. You adapted better growth, and because they're increased biomass, we think they just produce more toxin from that perspective.
So not even necessarily a strong evolutionary component there, just
really. No, unfortunately not. No. I mean, so the track that we took, and we published a paper on this year, is that we've more been interested in looking for diverse toxins and looking at diversity that's out there, and looking at natural diversity as a way to say, well, what part of the protein is actually important for activity? And can we as humans use our brains and figure out, well, okay, this yeast has a similar cell wall, but why is this killer toxin not attacking it? Well, obviously it's something to do with either binding the cell wall or the membrane. So we felt that more logical and systematic approach was maybe, the sort of the antithesis of Beacon, but that's the way we've actually gone. We've been looking at natural variation rather than evolved variation. So as our way forward, but honestly, I'm always thinking about how we could potentially adapt and evolve this system. But it's a hard problem.
Yeah, it does seem like you would need to have some robotic system that's just monitoring everything really carefully and responding appropriately, and selecting on a group level rather than an individual level.
Because we thought liquid culture would be one way to do it, but the problem is, if the killer yeasts, the idea would be you want to evolve a more effective killer yeast. So, but the problem is that in liquid culture, the killer yeast have to be a certain threshold to take over the population and win the fight, as it were. So if the evolution, if it's in favor of the strain that you're trying to kill, they'll just overgrow the killer yeast, right? So it's a very fine balance in liquid culture. In solid culture, it's a little bit better, because you can, when the killer yeast reaches a certain threshold, they will invade in solid culture in a structured environment. So, but that's not even that straightforward for evolving new killers. So I thought, well, what about yeast invasive growth? You can do that, right? You can have yeast that invasively grow, and you can have sort of these cultures that are mixing with each other and evolving in a sort of a semi-solid media. But the problem is that the semi-solid medias and don't particularly work for every time we had a killer yeast and we put it on semi-solid media, it didn't work. It didn't want to produce the killer toxins. So that didn't work either. So we've thought of a lot of things, but yeah, we haven't quite come up with something yet.
So one person you might be interested in talking with about this is Luis Zaman, who's at the University of Michigan now, who is building all sorts of robotic systems explicitly to try and do evolution of simple ecology, microbial ecologies with bioremediation goals and things like that. But yeah.
Great, thank you for that. No, that would be definitely somebody to hit up. I mean, yeah, the yeast ecology angle of killer toxins is fascinating. There's been a lot of work done on it. Cactophilic fungi are one of the primary model organisms showing how killer toxins can essentially cause a bias in the population composition of certain cactus rots. Killer toxin resistance arises quite commonly in those rots only to the killer toxins that are being produced in that specific rot. If you look at different populations in different cactuses, even, you see different community compositions that seem to be driven, they think, in some way by killer toxin presence. And there's been thought to be feeding preferences for Drosophila for killer yeast, which would help dispersal of the yeast, which could be a benefit, although there's no mechanism for that as of yet. That's work being done over in Europe. So yeah, there's the killer yeast ecology stuff is definitely, there's a lot of controversy as well, because some people think that killer toxin systems are addiction cassettes. So they get the virus, they get the satellite, and if they lose the satellite, they will be killed by their clone mates. But other people are thinking it's more of a beneficial system where you just get for interference competition. But I'm definitely in the interference competition camp, that's for sure, because we see a lot of evolution of killer toxins present also on the genomes of yeasts as well. We see a lot of duplications of these genes, telomeric amplifications of these genes, some signatures of positive selection going on. They're not strong, but still some elevated DNDS ratios. So yeah, that's on the genome. That's not on viruses. So we can see that as well going on.
So we're at time. We do have one question in the chat. If you could address that really quick before we wrap up.
Oh no, how do I do chat?
I can read it to you. It's from Mike Weiser. He says, so for
you.
diversity of toxins, are you looking at the chemical diversity of them or the diversity of the strains that they're able to affect with the toxin?
We're more interested in looking at the proteins themselves and the chemical diversity, so we can get a handle on, again, what part of the toxins are if we're looking at the family of K1s, which particular mutations in K1s are important for activity. But we are also we're actually discovering quite a lot of novel toxins too in this effort. So that's going to keep keep grants coming out for quite some time and papers some time. So we're finding a lot of new stuff. So thank you for the question.
Excellent. And thank you for the few who stuck around to the very end. I think some people had to leave at 4:30, but those of us who are left, let's give another round of virtual applause.
Thank you so much, all. I appreciate it.
Thank you for the excellent talk.
Oh, great. I have to go to lab meeting now, so I got to go too. So thank you so much for your attention. I really enjoyed this opportunity and the questions were great. Thank you again.
All right. Have an excellent weekend, everyone.
Good to meet you too. Thank you so much.
No problem. See you later.